Welcome to Mitosis.
I'm Alex.
And I'm Michael.
And today we're going to be talking about mitosis, which is important for all cell life. Did you know, depending on how long a cell's cycle is, mitosis could occur within 20 to 60 minutes. Discussing this topic will help further your knowledge on how the cell undergoes mitosis and how this affects other organisms. We are going to talk about all the stages of mitosis and what happens in each process.
Mitosis consists of 4 stages: prophase, metaphase, anaphase, telophase, followed by cytokinesis. To start things off, we're going to be talking about interphase. Interphase is the resting phase between successive mitotic divisions of a cell, or between the first and second divisions of meiosis. During interphase, the cell undergoes normal growth processes while also preparing for cell division. In order for a cell to move from interphase into the mitotic phase, many internal and external conditions must be met. The 3 stages of interphase are called G1, S, and G2. The first stage of interphase is called the G1 phase, also known as the first gap. From a microscope aspect, little change is visible. However, during the G1 stage, the cell is quite active at the biochemical level. In the G1 stage of interphase, the cell starts to grow. In the S phase, DNA replication can proceed through the mechanisms that result in the formation of identical pairs of DNA molecules, sister chromatids, that are firmly attached to the centromeric region. The centrosome is duplicated during the S phase. The 2 centrosomes will give rise to the mitotic spindle, the apparatus that orchestrates the movement of chromosomes during mitosis. In the G2 phase, the cell replenishes its energy stores and synthesizes proteins necessary for chromosome manipulation. Some cell organelles are duplicated, and the cytoskeleton is dismantled to provide resources for the mitotic phase. There may be additional cell growth during G2. The final preparations for the mitotic phase must be completed before the cell is able to enter the first stage of mitosis. In the graph above, mitosis and cytokinesis go together to make up the mitotic phase. The mitotic phase is the formation of 2 daughter cells that are exactly alike. After completing all the stages of interphase, the cell then goes on to mitosis.
The first stage of mitosis is prophase. Prophase can be broken down into 2 separate stages. The first is early prophase, then prometaphase. During early prophase, the nuclear envelope starts to break into small vesicles, and the membranous organelles, such as the Golgi complex or Golgi apparatus and endoplasmic reticulum, fragment and disperse towards the outer parts of the cell. The nucleolus disappears. The centrosomes begin to move to opposite poles of the cell. Microtubules that will form the mitotic spindle extend between the centrosomes, pushing them further apart as the microtubule fibers lengthen. The sister chromatids begin to coil more tightly with the aid of condensin proteins and become visible under a light microscope. Some key points of prophase are: chromosomes start to condense, mitotic spindle begins to form, and the nucleolus disappears. Once these things happen, the cell goes into late prophase, aka prometaphase. And this is what a cell looks like when it goes through early prophase. During prometaphase, which is the first change phase, many processes that were begun in early prophase continue to advance. The remnants of the nuclear envelope fragment. The mitotic spindle continues to develop as more microtubules assemble and stretch across the length of the former nuclear area. Chromosomes become more condensed and discrete. Each sister chromatid develops a protein structure called a kinetochore in the centromeric region. The proteins of the kinetochore attract and bind mitotic spindle microtubules. As the spindle microtubules extend from the centrosomes, some of these microtubules come into contact with and firmly bind to the kinetochore. Once a mitotic fiber attaches to a chromosome, the chromosome will be oriented until the kinetochores of sister chromatids face the opposite poles. Eventually, all the sister chromatids will be attached via their kinetochore to microtubules from opposing poles. Spindle microtubules that do not engage the chromosomes are called polar microtubules. These microtubules overlap each other midway between the 2 poles and contribute to cell elongation. Astral microtubules are located near the poles. aid in spindle orientation, and are required for the regulation of mitosis. For the cell to go into metaphase, it first must compact its chromosomes, break down its nuclear envelope, and grow its mitotic spindle to grab chromosomes.
During metaphase, also known as the change phase, all the chromosomes are aligned in a plane called the metaphase plate, or the equatorial plane, midway between the two poles of the cell. The sister chromatids are still tightly attached to each other by cohesin proteins. At this time, the chromosomes are maximally condensed. During metaphase, the mitotic spindle lines all the chromosomes in the middle of the cell, prepping for division. Before proceeding to anaphase, all chromosomes must align at the metaphase plate, and two kinetochores of each chromosome will be attached to opposite spindles. The cell has a special checkpoint to make sure it can go onto anaphase. This checkpoint is referred to as the spindle checkpoint. The spindle checkpoint makes sure that all the sister chromatids will split evenly. The dotted line here represents the metaphase plate at which all the chromosomes line up in the middle. In the picture above, you'll see what the end stage of metaphase looks like in a cell.
After the process of metaphase has been completed, the process of anaphase can now begin. During anaphase, which is also known as the upward phase, the cohesin proteins degrade, and the sister chromatids separate at the centromere. Each chromatid, now called a chromosome, is pulled rapidly toward the centrosome to which its microtubule is attached. The cell becomes visibly elongated, which looks like an oval shape as the polar microtubules slide against each other at the metaphase plate where they overlap. To sum up the whole process of anaphase, or to make it a little easier to understand, each sister chromatid is pulled towards opposite ends of the cell. For this to happen, the proteins that hold sister chromatids together is broken down, allowing them to be pulled apart. The microtubules that aren't connected to a chromatid extend to make the cell bigger. Now that the sister chromatids are separated, the cell can proceed on to the next stage of mitosis, known as telophase. In this part of the video, you can see where each individual sister chromatid is being pulled apart. In this diagram of anaphase, you can now see where the microtubules push poles apart. Also, you can see where the kinetochore microtubules pull chromosomes towards the poles.
After the cell has been through and completed anaphase, it then moves on to telophase. During telophase, also known as the distance phase, the chromosomes reach the opposite poles and begin to decondense, relaxing into a chromatin configuration. The mitotic spindles are depolymerized into tubulin monomers that will be used to assemble cytoskeleton components for each daughter cell. Nuclear envelopes form around the chromosomes, and nucleosomes appear within the nuclear area. In telophase, the mitotic spindle is broken down and two nuclei form, one for each set of chromosomes. The chromosomes, while in the nuclear envelope, then return to their original spaghetti-like form. After the cell finishes telophase, it then goes on to start the process known as cytokinesis. This is what the end result of telophase looks like. As you'll see, the chromosomes will be back in their spaghetti-like form, the spindle disappears, the nuclear membrane reforms, and the nucleolus reappears.
Although cytokinesis isn't technically a stage during mitosis, it occurs at the latter stages of mitosis. Cytokinesis takes place to divide the cytoplasm to form two new cells. After cytokinesis is done, there will be two new cells, both with a full set of chromosomes identical to the ones of the mother cell. The daughter cells will then live their cell cycle and might undergo the process that created them, mitosis. Now that we have walked through each stage of mitosis, I hope you will now understand the process and its impact on a cell. Mitosis is important when it comes to cell division. It replenishes our body with healthy new cells.
We would like to thank Dr. McIntosh for allowing us to make a video on mitosis for extra credit.
Thanks, Dr. McIntosh.